So welcome to your lesson on cell transport. Two major categories of transport are defined by whether you need energy to do it or not, and what direction you're passing things. So for example, passive transport, you don't need any energy, but active transport, you do. And the reason for that is that passive transport, the molecules move with something that we call the concentration gradient. Down here, this is meant to show you what a concentration gradient theoretically could look like, where you have a high concentration, so lots of particles in an area, versus an area where there's much less particles. And you will find that just due to something called Brownian motion, I've written the term here for you, because of Brownian motion, you've got the random collision of particles that begin to spread out. And when they begin to spread out, they head to where there's lots of collisions happening to where they have more space. And that'll just be a natural process that'll happen with a lot of particles. So for example, in a corner of a room, if somebody sprays an air freshener, it's very concentrated right when they spray it, but then over time those particles will spread out and fill the entire container because it's floating through the air, which is a fluid, so it flows to the other parts of the room. And eventually it may even be so thin that you can't smell it anymore. So another person walking into the room may not even be able to detect it because the particles are too spread out. We would say that they have diffused or diffused. So particles have diffused throughout the entire room, and they may or may not be detectable by your chemical scent receptors after that. But the point being that it doesn't take any energy for those particles to randomly move to where there's lots of them to where there's less of them. And that's really key. No energy required, moving with the concentration gradient. If, however, I want to go the other direction, I want to go from where there's few particles to where there are a lot of particles, so going from a low concentration to a high concentration, I'm going to have to force that to happen, and that's going to require energy to do that. So we would say that active transport requires energy because the molecules are moving against the concentration gradient. So now I want to give you a mental picture. I want you to imagine that you're working for the YMCA, and you have been put in charge of the kindergarten program. And on this particular day, it's gym day. And you're going to take all 50 of these little kindergarten kids and you're going to bring them to Eastview to go to the main gym, the big gym that has the double gym. And as soon as you open the door and you let those little kids in, do they stay in a nice pile right by the door? Nope. Instead, those little kids will do what little kids do best, and they will fill the space that they are in, and they will just spread out, and they will run everywhere, right? Until you establish some kind of order. So they're going to run everywhere. They're going to fill that entire space. So if you give these molecules, in our case, a space to move, they're going to move. You give the kindergarten space to move within gym, they're going to move. Now I want you to picture that it's time to take those little kids home. By the way, you as the leader standing against the wall, when you first opened the door and you just let them go, they were in a pile of kids. Is that a thing? They were in a little crowd of kids and then they spread out. So they went from high concentration to low concentration. That didn't require any effort on your part to make them do that, did it? You just stood against the wall and you let the chaos happen. Well, now on the other end of it, they've had their hour in the gym and it's time to pack them back on the bus. So now you're standing there and you're trying to get the little kids, but they're little kids doing little kid things. And every time you throw one into the corner, another one escapes. And you grab that one, you throw it into the corner. And you're trying to, with a lot of energy required, trying to keep them in one space. So you're trying to take them from where there's few of them and throw them in the corner where there's a ton of them all packed in. Of course, that requires energy. So if that's helpful as a mental picture, think of that as what's going on with these particles. You've got no problem moving from high to low. You just sit back passively and watch the process. No problem. But when it's time to go home, you have to pack them in somewhere. That's going to be an active process. You will be using energy to try to get those kindergartens back on the bus. Specifically, this type of passive transport, where it's going from high to low without any compulsion, is called diffusion. It's based on the random movement of particles. So not every particle in the high area is headed towards the low. Some of them could be headed back towards high. It's just random which direction they're going. It's just that overall, if you give it enough time, you will see it eventually spread out evenly in the container. It's somewhat inefficient if you are a cell that's trying to get a product either into you or out of you and you don't have a lot of time. Like you need the oxygen in order to run cellular respiration. And if the oxygen doesn't get there in time, you can't make energy. So you want to, under some cases, you want to be able to have some compulsion to move things across. But in this case here, there's no compulsion. It's just However long it takes is how long it takes. Now, if you have a really high concentration and a very low concentration, you'll see this move a little bit faster. That'll affect, the concentration will affect the speed of the diffusion. But if it's just a little bit over here and a little bit over there, then you're not going to be spreading out to the low area that quickly. So concentration does matter. It's a bit inefficient. You don't want everything to have to be transported in this way. You do want, under some circumstances, to be able to go from low to high, or to make sure that the high to low happens at a little bit of a better rate. So you could use a bit of energy to try to throw things in certain directions. It just depends what you're doing. So because of that, because some cells will actually have to rely on diffusion, something like a bacterium, very small cell, it's going to limit the size of their cells. Every bacterium, compared to a eukaryotic cell, which is what you have, is somewhere between 10 to 100 times smaller than one of your small cells. They are much smaller because they have less active transport going on, less decision of what they're throwing to one side and the other, and relying a lot more. And because their cell membrane is their actual surface area where they can do this exchange of stuff, they have to be much smaller. And of course, it means that you have to have lots on the one side and less on the other. What if you don't have lots on the one side and you still need that particular particle? Then you're stuck if you're just relying on diffusion. And it's only possible for the small uncharged molecules, things that can actually pass through the membrane, because the membrane has a selective process that it does. It's able to decide what comes in and out. In our case here, differentially permeable means the same thing as semi-permeable in this particular diagram. So remember that selectively permeable is referring to the idea that you have a choice. That's the bouncer at the door. Whereas semi-permeable is referring to if you're the right size and shape, or the right polarity, or the right charge, you can just go through. And in this particular example, it's just based on size. So you can see you've got water on both sides, and then you've got sugar shoved on the one side of this beaker, and then you have this membrane in between. And because there's a high concentration of water over here, and a low concentration of water over here, you're going to see water diffuse across the barrier. The sucrose in this case cannot move because of the selectively permeable barrier, so it's going to stay on this side. So osmosis is specifically the diffusion of water particles, whereas the other diffusion we just mentioned is any particle moving from high to low. So osmosis, the diffusion of water across a membrane. Now keep in mind, in this particular case, we have a semi-permeable membrane where the water is actually the only thing that's able to pass. So these little orange dots that are meant to simulate some version of solute, we could say sugar or salt or whatever it is, something floating in there, these particles are not able to pass through because the holes are not big enough for them. This particular semi-permeable membrane is based on size. Water's small enough, these guys just aren't. So we're not going to be changing the number of these larger particles, but we will for sure be moving water around. Osmosis can tend to trip people up because the idea of diffusion is simple, high to low, no energy. But the concept of osmosis appears to do something backwards. So I'm going to try to explain it to you in a way that gives you some confidence to know what the heck's going on with the water here. So I want you to consider, just from my little diagram here, we've got 1 part water to 1 part water, right? Equal parts. And then in my diagram, I've got 6 of these little pieces of solute to 3. So 6 compared to 1 if we're doing solute to water, and then 3 compared to 1. Clearly, there's more solute over here, right? Compared to water. If you give it a little bit of time, then you're going to end up having the water move over to this side. And what it's going to end up looking like is that the water is diluting the side where there's lots of solute. That's clearly what it looks like. So if at the bare minimum, that's what you remember, that water tries to dilute, that's great. But that's not actually technically what's happening. The water isn't moving over there because it wants to dilute anything. It's moving over there because it's following its own concentration gradient. It's where the water has a high concentration compared to the amount of solute. So if we do water to solute, 1 to 6 on this side versus 1 to 3, I hope you know that 1/6 is a much smaller piece of pie than 1/3. Okay, so if we're actually looking at the concentrations then of the water specifically, water is much higher concentration here than it is here. That's why the water will move from where there's lots of water to where there is a lower concentration of water. And keep in mind, we're not talking about the number of water particles here, because there's equal water particles on both sides. And clearly there's way more water particles here now than there is here. So it's not about the number of water molecules. It's about the concentration of that water. And the concentration is always how much water compared to how much solute, or how much stuff floating in the water that can't move through the barrier. Right? So water looks like it's diluting the part that has lots of solute. And you can certainly think of it in those terms if it makes it easier for you. But what water's really doing is moving from where there's a high concentration of water following its own gradient of water particles to where there's less concentration of water. I hope that sticks and makes sense to you. And it'll keep moving. Remember, this diffusion process is random which direction it goes. So you'll still have water molecules, even though some are heading towards the A side, you'll still have water molecules heading towards the B side and back and forth. But it'll keep happening. And even in this stage here, you'll keep having water molecules randomly crossing. But at a certain point, the number that cross back is equal to the number that crosses over. So you get these concentrations eventually become equivalent. You can see that 6 to 2 parts water, 3 to 1 part water, those are equivalent, because if I was to reduce those ratios, this would also be 3 to 1. So we would say then that the water molecules continue to move in both directions, both in and out of that area A, until something called dynamic equilibrium is reached. Dynamic means there's still changes, but those changes are balanced. So again, let's say 10 particles move that to the left, then 10 particles have also moved to the right. So there's an equal swapping going on. You're at dynamic equilibrium. So again, osmosis is the diffusion of water itself. And in these particular cases, the solutes, the stuff floating in the water, is not moving. It's only the water that's permitted to cross the barrier. These 3 terms are specifically trying to describe what's happening during osmosis specifically. So you have to be talking about the movement of water, and what we're going to do is describe which direction water moves based on the concentration of the solute. So for example, in the last picture, you had the water move from where there was a low concentration of solute, but a high concentration of water, to where there is a high concentration of solute, but a low concentration of water. So it's the water, again, still moved with its own diffusion gradient from where there was lots of water to where there was less in terms of concentration. But it sure looked like it went from where there was less solute to more solute, doesn't it? The term that we use for same, equal number of water particles compared to solute, so equal concentration, is isotonic, same. But when we're talking about having a higher concentration of the floating stuff, of the solute, we would call this hyper, and we would call this hypo. And the word tonic is just referring to the solute that's floating in the water. So keep in mind, we're talking about the concentration not of water, but of the other thing. So where it is a hyper concentration of solute, it's actually a very low concentration of water. And where it's a hypo concentration of solute, it's a very high concentration of water. That's why it's always going to appear to move from where it's hypotonic to where it's hypertonic. So you're going to see that water moving across the barrier naturally like that. Let's talk about the parts of the word. So iso is going to mean same. We've already seen that word iso before when we were talking about isomers, right? We meant that they had something same about them. They had the same parts, just in different locations. So here we have the same tonicity, or same concentration. Okay? That's what that refers to. Again, it doesn't mean equal number of particles. It means equal concentrations. Because clearly in that diagram above, clearly we can see it's not equal water particles, and it's not equal solute particles, but it is equal concentration. So again, this would be considered isotonic. Same on both sides of the barrier. If it's hypertonic, it means that the concentration of the solute is very high. Hyper means more, hypo means less. Can you think of other times you've seen the word hyper? Hyperthyroid. And what does that mean? Yeah, so hyperthyroidism, you're producing too much of a hormone that's causing your body to speed up the cellular respiration in your body. We'll learn about that in our last unit. Another hyperextension, which means, okay, when you bend your joints farther than they should be going, you hyperextend them. You're extending beyond what's natural. That's how you end up blowing a knee or blowing an elbow, but hyperextension. Hyperactive, you've heard of before. Much higher activity level than what's normal. What about hypo? And please don't say hypopotamus, because that's not what we're looking for here. Hypotension, which means low blood pressure, lower than normal. You could also have hypertension, which is high blood pressure. Hypothermia, less heat than you should have. What about a hypodermic needle? Derm is your skin. If it's a hypodermic needle, it means it goes under the skin in order to deliver its payload. So you've seen these words before. You've even seen it when you were studying math back in grade 9, back when Pythagorean theorem consumed your life, and you were looking at the hypotenuse of a triangle. Hypotenuse, it's two words meaning below and stretched between. Tenuse is like the word tension. You may not know this, but you've got the hypotenuse, which you know is the one that's opposite the right angle. But the two parts of the triangle that make up the rest of it are called legs. So the idea is that you've got the hypotenuse stretched between the legs of the triangle. That's why it's a hypo. It's below the two legs, tenuse, stretched across. So you've seen these parts of language before, hyper and hypo. So hopefully that'll jog your memory when you try to remember which one's which. Hyper, more, hypo, less. But more and less of what? It's more and less of the stuff that's floating in the water, not the water itself. And that's the part that usually messes people up. So if the cell is hypertonic, then keep in mind you've got a barrier, which is in this case the cell membrane, but you've got the inside which is of the cell, which is being described as hypertonic. But that means that there's a lot of solute inside, then the other side of the barrier must be compared as having less. So if one side of the barrier is hypertonic, the other side of the barrier is hypotonic. Again, like the picture above. This would be described as hypertonic. This would be described as hypotonic. Just because you have to compare it to each other. So once again, if the cell is described as hypertonic, it's because the other side of the cell membrane, what it's floating around in, must be hypotonic. And vice versa. If the cell is hypotonic, then it means the other side of the barrier is hypertonic. It's always a relational term. The only time that it's the same is if you're talking about isotonic, which is what's shown in this first diagram here. So here we have our representation of a cell floating around in some fluid. And because the concentration of this solute is the same inside and outside, we would say that it is isotonic. Same concentration is literally what the word means, isotonic. Then you've got a cell that has a high concentration of solute on the inside, and the environment has less, so the water will move in. And again, it's not because the water is moving from low to high concentration all of a sudden, although that's what it appears to be in terms of the stuff floating in the water, but because there's a lot of water compared to solute here and a lot less water compared to solute here. So it's following its own concentration. But it sure looks like it's trying to dilute. So the water will always move from where it's hypotonic to where it's hypertonic. And we see the same thing in the other situation. Here we have a hypotonic cell, hypertonic environment, and the water is now moving out. It's seeking to dilute the hypertonic environment. So just keep that in mind. It always moves from the hypo side to the hyper side. That'll help you when you're trying to analyze your lab and also give you good terminology for when you're trying to deal with things on the tests. Just as I've said before, if you want to get good at these terms and you want to really be able to understand what they mean, like any new language, this is the English language that you're learning, but like any new language, you need to use the terminology, otherwise it won't stick. So from here on out, be thinking about the concentrations in terms of hypertonic, hypotonic, isotonic, so that you can try to understand what water will do. Let's just consider the animal cell first. Remember that we are going to be considering the cell floating around in some kind of fluid that's outside of it. So if we're going to describe the solution it's floating in or its environment as being isotonic, then the cell is also isotonic. If the environment is hypotonic, the cell must be hypertonic, and vice versa. If the environment is hypertonic, more stuff floating in it, then the cell would be hypotonic. Okay? So again, it's a relational term. So isotonic, isotonic, hypo, hyper, hyper, hypo. But now we're going to consider when the water does move, what will happen to the cell itself. Now, obviously, if it's the same concentration inside and outside, of course, we're going to be already at dynamic equilibrium. We're going to have water molecules move out, but the exact same number will move in. So overall, the concentration remains the same. This is perfect for an animal cell. And you'll see why in a second. If the cell has a higher concentration of particles and solute inside, so that the cell is hypertonic and the environment is less concentrated, then you're going to have the water move into the cell, the water moving into the cell, and that cell will continue to swell as long as the water moves, and it swells and it swells and it swells and it swells, and eventually it reaches literally a breaking point if you continue to add water, and that means it'll swell till it blows, and that idea of the cell bursting is called cytolysis. Cyto means cell, and lysis is the idea of bursting or breaking apart. So cytolysis, the cell bursts because it was too hyper to begin with, and then as the osmosis began to happen and the water moved in, it just couldn't contain all the water that came into it. Boom. If it's the vice versa, and you have a hypo cell versus a hyper, so a salty environment, not so salty cell, the water will move out to dilute the environment, and the cell will shrivel up. Also not ideal. The cell can't shrivel like this, otherwise all its functions stop working. So cytolysis is bad when you blow it up, but crenation, when you shrink it or shrivel it, is just as bad. That's why we would say that animal cells need to have an isotonic environment. So let's say you have surgery and you've lost some blood fluid, and so they need to give you an IV to just, even if you're just dehydrated, they'll give you a saline solution. Saline just means salt. They have to make sure that the solution that they pump into your body, that liquid that goes in there, has to be the right concentration. If it is too salty, you have this problem. And the fluid that you're throwing into your blood will then shrink your cells. Not good. You don't want to dehydrate your cells. And if it's vice versa, if it's not enough salt in the saline solution, then you're going to have this situation where the water from the new saline solution will then fill up your cells and could potentially lead to cytolysis. So either way, those saline solutions being pumped into your body have to be isotonic to the normal human body. Okay? Not too much salt, not too little salt. And they have to double and triple check the bags before they leave the factory where they're created, before they get sent out to the hospital, because you can kill a person this way. It's very, very important that it has the perfect concentration. It's also why, if you were to flush your eyes out with water versus use the eye drops, it's why regular water could burn. It makes your eyes feel like they're burning. It's because you have this situation. The regular water doesn't have as much salt in it, and so you have the water moving into your eyes causing inflammation. That's why they get red and swollen. Whereas if you have eye drops, the eye drops are isotonic to what the normal human eye should be in terms of saltiness. And so then you would have a normal situation. You're not shrinking or blowing up your cells. You have a nice, comfortable situation. So even that, you have to have a high-quality eye drop in order to make sure you're not damaging your eyes, drying them out, or swelling them. Plants have a different situation. They have a different idyllic situation as well. And that's because they have two parts that the animal cell does not have. The first part is the water vacuole itself, the tonoplast, if you will. And the second thing is a cell wall. So that if it is isotonic, then you have a condition that's called flaccid. The first C makes a K sound, and the second C makes an S sound. So you'd say flaccid, or flaccid if you want. In this case here, iso, you actually don't have an ideal plant cell. What you want is this one down here called turgid, or to have something called turgor pressure, because plants don't have a skeleton. You've got bones that can hold you up. Plants don't have bones to hold them up. Instead, what they rely on is these boxes, these cellulose boxes, the cell walls, need to be full of water, kind of like how we've shown in this picture here, so that the water vacuole pushes up against the cell membrane, which pushes up against the cell wall, and pushes pressure on it to keep that box nice and firm. This is ideal. You want it to be turgid, so water is constantly moving into the cells and pushing, pushing, pushing. Versus if you were sort of not right pushed up against it, you'd have a bit of a floppy box. I want you to picture a cardboard box with a water balloon in it. Now I want you to picture you've got a whole bunch of these boxes stacked on top of each other, and then you try to sit on them. If the water balloon is empty, you're going to sit on the boxes and collapse them really easily, aren't you? Well, that's the case that's down here. If the water balloon is empty, the vacuole is empty, it's no longer putting pressure on the sides of the box. You've got a pretty weak box. You're going to crush them. This is when you see wilted plants. And the leaves droop and the whole parts of it droop. It's because they're dehydrated. You knew they were out of water. Obviously, they looked like they needed water, but this is why. They're no longer able to hold their shape because they're empty of the water that they need to be full of. If a plant is healthy, it's able to lift its branches or whatever, depending on the type of plant, and because it's turgid. If it's flaccid, this is when it's not so wilted that it's necessarily dropped all of its leaves down, but it's sort of in that in-between world where it's not as standing up as it should be. The boxes are not as full as they should be, so that water balloon is not completely pressing against the sides. It's giving you enough that you're not going to completely collapse the boxes, but you're certainly not going to be giving enough pressure to keep the boxes strong and upright. Flaccid, not ideal. Flaccid is when it's isotonic. Turgid, perfect for a plant. In order to make it turgid, you have to have a hypertonic environment. So you need to have lots of salts and whatnot stored inside the water vacuole so that you'll encourage water to constantly be coming into the cell. So again, the water vacuole, called a water vacuole, it's not just water in there. There's tons of things in there, lots of solutes in there to draw water in and keep it nice and stiff. And then if you want to move water out, then you take the solute that's in there and you throw it outside of the water vacuole. Water vacuole will shrink to follow the solute. Plants will actually choose when they want to be completely turgid and when they want to be a little bit more flaccid. I'm sure you've all noticed that plants are actually able to move. Not in terms of changing their location. Once they're rooted. But I think you know that they're able to turn their leaves towards the light, and they can move flowers around. They can open and close flowers. They're able to lean into the wind so that they don't get knocked over. They're able to do all that kind of stuff. And the idea would be that if you want to, let's say you want to turn to the left, so you're bending to the left, I'm trying to describe for the online people. If you're bending to the left, it means you have to pump up the cells on the right and then reduce the size of the cells on the left so that you can actually bend to the left. And if you want to do the other way around, then you pump up the cells on the left and you reduce the cells on the right, pull the water out of them, and then you can lean to the right. So that's how they can actually move their leaves around and be able to change the direction of things. You just pump up some cells and pump down other cells. So they can actually, on purpose, make some cells more turgid than the other. How they do that? They do that by throwing salts. There's all different types of salts. They're throwing salts on one side of the vacuole or the other. You want it to fill up, throw the salts inside, the water will follow to dilute. You want to pump it back down again, throw the salts away, the water will follow and it'll shrink it. One more term just to mention here. If it is specifically a red blood cell that has burst, although technically it's a type of cytolysis, it's a cell bursting, we would be more specific and call it hemolysis or hemolysis if you want to try to remember how to spell it. Hemolysis. So one more time with higher quality graphics than I could provide on the previous page. You have your animal terms, crenation, which means they are shrinking, or cytolysis, which means they are swelling and possibly bursting. Again, if it's a red blood cell, RBC, then you would use the word hemolysis or hemolysis. Ideally, for an animal cell, you want it to be isotonic. Anything other than that will cause these conditions. Memorize the conditions that'll cause that. In terms of plants, flaccid would be the isotonic condition. Turgid is when you have high turgor pressure. That's the water vacuole pushing against the sides of the cell wall. This is ideal. This is what plants want. And then plasmolysis is when you shrink that plasma. You shrink the amount of water inside the water vacuole so that the box collapses. And if that's the case, it's going to be wilted. So very wilted, will likely die. It depends on the species. Some species can handle being plasmolyzed for a little bit longer than others, but most likely this plant is about to die. Flaccid is ever so slightly wilted. You still probably have time to save the plant. And then ideally, nice swollen water vacuole pushing against the cell membrane, which pushes against the cell wall, nice and stiff. Has turgor pressure. Just a reminder, passive transport refers to no energy required because you're moving with the concentration gradient from high to low. So we talked about just the diffusion in general of small particles that are able to go from one side of the membrane to the other, no energy required, they're just flowing through. And that's through the actual membrane itself. Then we said, well, then there's osmosis, and we have to consider not just the concentration of particles, but the concentration of the water itself, and it being able to move from an area where it's hypotonic, less particles, to an area where it's hypertonic, more particles compared to water, but the water's still moving with its own concentration gradient. In this conversation, we're still talking about diffusion, still a passive process, but what we're acknowledging is that some particles are too large to fit in the gaps between the phospholipids. So they can't just flow through. They're too big. They're going to bump up against the phospholipids and not be able to go any farther. And that's why we have to have these integral or intermembrane proteins built in. It's still diffusion, but now we're going to give the larger particles some help. We're going to facilitate their diffusion, help them across the membrane. So the particles are too large to cross. The selectively permeable membrane are assisted by these proteins. And again, still moving with the concentration gradient. I still have to have more of them on the one side than the other. I'm still moving from high concentration to low concentration. And there's still no energy required. That's really key. This is still a passive process. Any description of diffusion must be passive. So what do we have? Well, in one case, we've got something called a channel protein. And basically, it just opens up a space between the phospholipids that certain particles are able to go through. Now, it doesn't just mean that if you're of a certain size, you can get through, although there are some channel proteins that would be like that. It could be that maybe the channel itself is negatively charged. So it would attract positive molecules to come through, but it would repel the negative ones. So you still have to be the right size, shape, polarity, all that kind of stuff. That still has to all be met as your criteria. But if the channel is designed for you, you as the particle are able to pass through from where there's lots of you to where there's less of you. No energy required. The other type is called a carrier protein. And this is a little bit more complicated, but still doesn't require any energy. So for example, this particle here could come in, and then because of the hydrogen bonding that could happen and all these different side chain interactions that could happen with the protein, I might be able to come in and cause that protein to change its shape ever so slightly. So that in the next second, I've actually been locked on this side, I change the shape of these proteins, and now they open me up and let me drop into the other side. So that's a possibility as well for the carriers. So we can open it partially, molecule enters, the protein changes shape, and then the molecule gets released to the other side. So more like you walk into, like the revolving doors, you walk into the door moves, and now you're free to go to the inside. Then if you want to come the other direction, you have to go into another door, it turns, and you're released to the other side. So there's a double step there. But in this case, again, it's passive, no energy required. It's just the nature of the molecule itself was able to trigger the change in the shape. So not every molecule will be able to trigger that change, which means that this carrier can be specific to certain types of molecules. You have to be the right molecule in order to mess with the protein, in order to change it so that you can get through. In other cases, the carrier can actually have the molecule attached to it, and then that changes the shape, and it literally grabs it and flips, and it throws it into the inside. And then in order to be rebooted, you have to have another molecule come the other direction so it can throw it out, and so on and so forth. So you can grab and flip, or partially open and have this conformational change and allow entry to that particular molecule. It's particular. And again, it's facilitated diffusion, moving with the concentration gradient, no energy required, but they need a little help to get through because they're too large. That's the end of our conversation on passive transport. Now we have a short conversation on active transport. The first type of active transport that we want to consider are the active transport pumps. This will become very important when we start to talk about nerve transmissions or how the kidney works and things like that in our homeostasis unit. So we'll come back to the active transport pumps. For now, what you need to know for our beginner unit is to know that you have to have cell energy called ATP to run it, and that it's against the concentration gradient. All types of active transport require energy and are against the gradient. So what we're doing here is grabbing the kindergartens from the general gym and packing them into the corner. It's going to take us a lot of energy on our part. In this particular one, this is a very famous example, the sodium-potassium pump. What we're going to do is have a pump that actually works in both directions, and it pumps two things that are very important to me within kind of at the same time. So the one particle is going to activate it to go the one direction, and now that it's open to the other side, it's going to be activated by a different particle to throw them to the inside. So for example, these orange things represent the potassium ions. Let me see where we can start here. Let's actually start up here at this picture. In this picture, you have the sodium or the little green, and they will come into the pump. By coming in here and then by adding a phosphate group, we're going to learn more about ATP in our next unit. It has 3 phosphates attached. It's a TP, so triphosphate. And this is DP, diphosphate. So we steal a phosphate off the ATP, and when you attach it as a functional group to this protein here, it actually changes the shape of the protein, and then it'll pop these guys out the other side. So the change of shape happens. It opens the gate for these green guys to move to the outside world. Now that the gate is open to the outside, now the potassiums, which are quite a bit larger, they're able to come in and fill in that space. The phosphate gets ripped off, changes the shape. So here they are here. The phosphate gets removed, which changes the shape back to normal and releases these guys to the inside. And now it's ready again for these guys to move in. So you have them move in, add the phosphate, change the shape, release them. These guys come in, remove the phosphate, release them. Change. And that phosphate being added and removed is where we say that's the energy that's required. ATP is a type of cellular energy. Without that ATP, you never get the first change to happen. That's why we say it requires energy. Okay? And we're probably pumping from where there is few sodium to where there's already a ton of sodium out here, outside the cell. And vice versa. We already have a ton of potassium inside, so we're taking the potassium that's outside, low concentration of potassium, and putting it to where there's a ton of potassium on the inside. And again, you're going to see this in our nerve transmissions. This allows us to send an electrical message all the way down a nerve cell in order to then trigger the next nerve cell to send a message from the body to the brain, from the brain back to the body. For now, what I want you to remember is that sodium-potassium pump is a type of active transport pump. If you can also memorize the details that it's 2 to 3, great, but that'll serve you better in our future units. So it depends how much space you have right now in your brain to try to memorize all this stuff. Okay, so active transport pumps is then our first type of active transport, again, with energy, ATP, and against the gradient. 2 other types of active transport, again, active because it's against the gradient and it requires energy, I'm going to say that a million times so it sticks, is something called exocytosis. If you had to guess what direction I'm moving objects, which one would this be? In or out of the cell? Out of the cell. Exo, I'm exiting. And cyto again means cell. So when you see a cis, an osis like that, that basically means the process. So this word literally means the process of moving stuff out of the cell. That's what it literally is defined as. Okay? So what we're doing here then is something called secretion. So either I've made a product and it's been code from the DNA was transferred to an RNA molecule. It left the nucleus through a nuclear pore. A ribosome clamped on, either free-floating or attached to the endoplasmic reticulum. I read the RNA code, so the nucleic acid code. I created an amino acid chain out of that using my ribosomes. Now I've got a polypeptide. The polypeptide gets folded. It gets transported by the endoplasmic reticulum up to the Golgi body here through a vesicle. Then the Golgi body will then modify it, make sure it's a fully functioning protein. Then it will create a vesicle, something like this, called a transport vesicle. And then the vesicle will then move out to the cell membrane and bind to it. So that you can see that this part here is this part of the vesicle. It just becomes part of the cell membrane. So these vesicles are going to actually literally be enlarging the cell membrane at this point because I'm adding more and more material. They're made of the same stuff. It's a phospholipid vesicle. So it comes and it releases, and when it opens up like that and releases, whatever is inside of it is going to be dumped into the extracellular fluid, and then either that goes into the bloodstream or it influences the cells nearby. It just depends what type of particle it is. So that's it. That's exocytosis. You use energy to change the shape of the cell membrane, and you're just constantly dumping stuff out. Again, manufactured products, stuff I want to dump out, plus any garbage that I don't want to keep inside. The next one is endocytosis. What direction am I probably transporting things? To the inside of the cell, so things from outside to inside. Now, there's going to be a couple different terms because we're trying to establish what size of the thing are you bringing in. Is it a small dissolved substance that you're trying to bring in, or you're going to do the exact opposite of what the exocytosis was? Instead of having a vesicle come to the surface and releasing it, instead you're going to have the surface itself, the cell membrane, will pinch in, kind of like you see in these 3 stages here. It will pinch in until the phospholipids meet each other, and then the solute particles that were on the outside get captured, and a full vesicle gets formed, and now it can go meet up with a lysosome who can digest it and do whatever it needs to do with those parts. Endocytosis, specifically penocytosis, is the small dissolved substances, and this is an actual electron micrograph of what it really does look like. You actually see these little vesicles forming and moving into the cell. Then if you have something larger, maybe it's not dissolved, floating around the liquid, and you still need it to come inside. Peno means to drink, so it's the process, the osis, the process of the cell drinking particles, versus phagocytosis, which is the process of a cell eating. Phago or phago, depending on your preferences. American versus British background, I guess. So this is just taking in larger things. And again, it's form a vesicle out of the cell membrane, and that creates this food vacuole or whatever other type of vacuole. And now the lysosome can do its damage on it. This is how white blood cells operate when they're trying to get rid of dead or damaged cells or trying to attack bacteria. They'll do exactly this. The bacteria would be this cell rather than a red blood cell here. The bacteria would be here. It would form this little vesicle around it, and then the lysosomes inside the white blood cell would destroy it, rip it to pieces. There's a few more steps that would happen with immunity, but that's it in general. So we have these macrophages, they're called. Macro meaning large, phage meaning the eaters. So these white blood cells that are out there called macrophages, and their job is to go and eat and destroy any of your bad cells, damaged cells, or any foreign material like a bacterium. The final category would be receptor-mediated endocytosis. So it's the process of bringing something into a cell, but it has to be triggered by a receptor first. It doesn't just automatically get sucked into the cell. So in this particular case, you have a particle that has to match what's on the outside of the cell. So remember we said there was glycoproteins and glycolipids and different types of proteins that are just peripheral, they're on the edge, and they're used for cell communication and all sorts of things like that. So a receptor could be a protein that has been embedded in the cell membrane, and when the right particle with the right kind of key comes along and triggers the receptor, that will trigger the process of pulling it into the cell, creating the vesicle, and now it's inside and I can do what I need to do with it. I can even separate that out so that the particles then can go and be used, and the receptors I can reuse themselves as well and put them back into my cell membrane. I just have to put the vesicle back up. So you pull it in, you do whatever you want with the contents, and then you put the vesicle back. It receptor mediated because you have to trigger it. And that, my friends, is the end of our cell transport lesson.